Now, yeah, I'm done. Yeah. Thank you, sir.
Yeah. Did anybody not get a copy? Okay, it's coming. Okay, my plan is to go through this question by question, hopefully giving you some test-taking strategies that you might not have thought of along the way, although if you've taken any multiple choice exams at all, you probably figured these things out. I remember a guy I was talking to who did not speak Russian, yet he passed a CLEP test on Russian because he knew how to take exams, multiple choice exams. Couldn't read a thing on the exam. But it worked. So it's conceivably possible to be able to do these things just on test-taking strategy. Of course, be aware that I know that test-taking strategy, so you know, it's a little mind game between you and me. No, I'm just joking. I don't think about these things when I write these exams, and besides, at least half to three-fourths of the questions come from a test bank anyway, so they haven't thought of those things. The exam questions, this program will scramble the answers for the questions so that they're never the same twice. So that's kind of neat. Do expect to see at least two different forms of the exam. Because this is such a crowded room, what I tend to do is alternate forms of the exam. We typically have them in two different colors, so you can figure out which exam you've got. Also, remember to bring a number 2 pencil. I will bring the Scantrons. For those of you at home, you'll be marking your exams directly instead of using a Scantron, so don't worry about it. But for you in class, you need to bring a number 2 pencil so that the Scantron doesn't screw up on your exam. And do remember also, if you have a lot of erase marks, the machine doesn't like those, and you probably will want to fill in a fresh form towards the end if you get finished and you have lots of erase marks. Okay, well, let's go ahead and go through this exam. Hopefully we'll be able to see this. It's a little bit iffy. I expanded the lettering a little bit on these things so that we could see it a little bit better, so I hope it'll show up. This first question, members of the kingdom Fungi. Now, when you're going through questions like this, I guess the first thing that you do, sort of, is you look at the answers and you try to decide which ones could possibly be correct. So, you know, you're reading this thing and you say, well, yeah, yeasts are fungi, so that's correct. Mushrooms are fungi, so that's correct. They decompose the remains of dead organisms and absorb nutrients from the leftovers. That's correct. So we're sort of in a quandary here. We know that there's at least 3 answers that are potentially correct. If you've done your reading, you know those are true, the kingdom Fungi. So you have to find an answer that incorporates all of those. The only one that incorporates all of those is D. Right? So that must be the correct answer. But just in case, you'd want to look down at E, and you'd look at that and you'd say, well, do they use photosynthesis to produce their own food? No. Members of the kingdom Fungi cannot do that. Right? By definition, they cannot do that. So D is the correct answer. A lot of times you'll see if you had A, B, and C all being correct, and of course this answer might be just be listed as, you know, A, B, and C are all correct. You see that quite a bit. When I'm writing questions, that's what I prefer to do because I don't like to retype things a whole bunch of times. Okay. But those can be confusing. I remember several years ago, must have been 4 or 5 now, there was a guy who came up to me after an exam where I had a lot of answers where he had to pick A and B or A and C. And he got really mad. He said I had designed that exam to fail people, and I didn't feel that way. It's just if you know the material, then you know which answers are correct. This one's fairly easy because it lists all of these. There's another little piece of test-taking strategy here. The longest answer is often the correct one, strangely enough. So if you have no clue on a question, you might want to pick the longest answer, because your odds are a little bit better on that than otherwise. Okay? I mean, that's really true. The longest answer tends to be the correct one. Another piece of test-taking strategy. A is almost never the correct answer, especially if it's a human-written exam instead of machine-written exam. The reason is because a lot of times if an instructor is writing an exam, they'll try to make sure that there's not a lot of A's and they overcompensate. So there's rarely A is the correct answer. So if you're also clueless on a question, you know, you can usually eliminate A, unless it's the longest one. Now, I didn't say this test-taking strategy would be valuable, did I? I didn't say I'd give it to you. Okay, scientifically valid, a hypothesis must be. So, you know, this is hearkening all the way back to Chapter 1. You're trying to figure out what a hypothesis really is. If you remember from Chapter 1, a hypothesis is basically an educated guess about a particular phenomenon or observation. We often phrase those as a question, but they don't have to be phrased as a question. You know, what's causing this phenomenon? Well, really, your hypothesis would be one step below that question. All right, so certainly we hope that B is not the correct answer, because if something is based on faith, it's not science. All right, it's that simple. So we look at the third one, testable. That's correct. Falsifiable. The general argument is that they need to be falsifiable. All right, so both of those are accurate. So then we'd pick E as being the correct answer. Testable and falsifiable. Okay? All right, which of the following statements is true of the domain bacteria? So you just have to go through the statements and try and figure out which one is correct. Okay, well, you know, if you remember the distinction from Chapter 4 between prokaryotes and eukaryotes, right? You remember the characteristics of prokaryotes, of which bacteria are members. They lack organelles. They lack a lot of membrane-bound internal compartments. And one of those compartments that they lack is a nucleus. So you can eliminate that answer. You know the bacteria are unicellular, so you eliminate that one. They do not eat other organisms. They take in simple nutrients. Right? And so then you pick D, and that's the correct answer. See, this is one of those where the common test-taking strategy would get you the wrong answer because the longest answer isn't correct. Right? In fact, it's the shortest one. Okay, any questions on those so far? Make sense? Good. Okay. On to the next. Which of the following is or are true about evolution? Answer A is actually a very, very common misconception about evolution. A lot of people think that evolution is a passive process, that it is somehow the chance appearance of new traits, and it's the chance choosing of those particular traits for particular environments. But they forget that there is a process going on that picks among all of those different variations, and that's natural selection. All right? So evolution is not a passive process. It is a process in which natural selection picks the traits that are most valuable for reproductive success. All right? It's not a chance process except in acquiring the variation in the first place, as we'll talk about next term. Okay? Individuals evolve. You sort of could say that. But it's really a species or a population that evolves over time as traits change in frequency. Okay? Organisms evolve structures because of need. Okay? No, they don't. There is no need that's there. If an organism actually happens to have a trait that's valuable, then it persists, okay, in that individual's offspring. Evolution is deliberate and purposeful. No, there's no purpose in evolution at all. Some people may ascribe a purpose to evolution in order to rationalize some other things that are going on in their minds. It is not a deliberate, purposeful process. See, I would actually argue in looking at this, and what I did is I just pulled these out of a test bank, and this is a question that I would throw out. And the reason is because I don't believe there's a true answer there. I do not believe there's a correct answer in that question. If you had a question like this, turned out you had a question like this on an exam, then what I would simply do is throw it out and give everybody 2 points for that. Okay? Yeah, this is a question that I would argue does not have a correct answer. Right? Because I think what they're after is evolution is a passive process. It's not, because natural selection drives evolution. Okay? It's not passive in that sense. Okay? So this question would be a throw-out. And I have no problem throwing out questions like that. Okay, number 5. In a DNA double helix, what does adenine pair with? I hear a lot of answers being bandied about. This is good. This is making you think. To answer this question, you need to remember the 2 simple rules about base pairing in DNA. C's with G's, A's with T's. All right? A with T, C with G. Remember that, you can't go wrong. Now, it's a little different in RNA, which supposedly is single-stranded, but occasionally RNA will be double-stranded. In RNA, A can pair with uracil. But we don't have to worry about that for this question because this is DNA. Right? So adenine is always paired with thymine. Okay? So this one is just a brute force memorization question. You recall the fact. I try to mix up my exam questions so that there's about half of those kinds of questions and about half of questions where you actually have to apply that knowledge to solve a question. All right? The application, the conceptual problem solving, the critical thinking skills are the most important thing you can get out of this class. The facts you can look up, except on an exam. Acid precipitation. My thought on this question is that A is probably the correct answer. I mean, that makes sense, right? It's certainly not due to burning rainforest timber. That's bad, but that produces CO2 and contributes to global warming. Right? And it's being done at an alarming rate. One of the problems with using rainforest land for agriculture is that it's some of the crappiest land in the world. You'd think you got all these plants growing, it's lush, it's wet, there's got to be tons of nutrients in that soil. It's not. It's been leached. It's the most nutrient-poor soil in the world. So, you slash and burn that rainforest, you start agriculture on it, and you can only get a decent crop for a year or two before the nutrients are depleted. So what people have been doing is, instead of trying to adopt alternate farming techniques for that land, they simply slash and burn some more rainforest and move on. So we're losing rainforest at an alarming rate, and that's increasing the level of CO2 in the atmosphere. But that's a different issue. Defined as rain having a pH above 7.4. That's not the case, because above 7.4 is basic, not acidic. Okay. Damages natural wilderness areas but does little or no damage in towns and cities. That's incorrect as well. You remember that picture I showed of that statue in Germany that had been almost completely obliterated by acid rain from industrial sources. Mostly coal-fired plants. When you burn coal, you release a lot of sulfate in the air, and that turns into sulfur dioxide. Say that again? Yeah, actually, I wouldn't be surprised. The question is, isn't that the same kind of thing that's ruining some of these ancient monuments like the Parthenon in Greece? I wouldn't be surprised. I don't know for sure, but it wouldn't surprise me. Okay. All right, so if I was giving an answer for this, I'd say A. That makes sense. Okay. The acid in the snow remains in prolonged... Yes? Doesn't precipitation mean rain? No, precipitation means water from the sky. Who cares if it's frozen? Yes. Good question. Thank you. In fact, that's why they say precipitation instead of rain.
What is the deal with the glaciers that freeze and have pure water?
Oh, yeah, then what's the question about the glaciers that freeze in their pure water? Well, that's dissolved salts, right? Glaciers start out as seawater, and there's a bunch of dissolved salts in them. When that water freezes, then you get those salts that are released. Okay, so it's a different issue. Okay, which one of the following represents the sequence of organization from simplest to most complex of actin? This asks you to directly recall a figure from the book, but you don't necessarily have to if you remember the hierarchy of molecules. There's a figure in Chapter 2, I think it is. It's either Chapter 2 or start of Chapter 3 that shows these. Okay, so you just got to go from the smallest to the largest. You can eliminate two answers immediately, right? C and D can be eliminated because those aren't the smallest. They don't start with the smallest. So you can eliminate those. Now you're stuck with A, B, and E, and you got to figure out which one is correct. All right, so actin is a molecule. The myofibril is an organization of actin and other molecules, actin, myosin, and a few other proteins. Right? The muscle cell is packed full of myofibrils. The muscle cells are organized into muscle tissue, and the muscle tissue is then organized into an organ, the flat muscle. So E is the correct answer. This one organizes them from simplest to most complex. The problem with A is that cell is before tissue, and here you've got, let's see, actin, myofibril, tissue before cell, so you've got to flip those. All right. So E is the only correct one. All right, this one should be easy. The nucleus of an atom contains. Hopefully that's a gimme. Okay. Got to be protons and neutrons. It can't have only neutrons because you don't know of any that have only neutrons. Even the simplest one, hydrogen, has a proton and no neutron, so there's a proton there. The next one, helium, has 2 protons and 2 neutrons. Even deuterium, which is an isotope of hydrogen, has a proton and a neutron. You don't get neutrons by themselves, interestingly. I think the half-life of a solitary neutron is something like 10 minutes. They don't last very long. Okay, so that's got to be the correct answer, protons and neutrons. The electrons are outside of the nucleus. All right, next one. Okay, you put a teaspoon of instant coffee and a teaspoon of sugar in hot water. All right, so which of these statements are true about that thing you've just made? I haven't used these terms in class, and so this comes directly from your reading. Solute is what's dissolved, and a solvent is what dissolves the solute. Okay? So a solute is what's dissolved, and a solvent is what is dissolving the solute. Right? So in this problem, the hot water is the solvent, and the other things are solutes. All right, so now you know that B and C are not correct, but you knew that already because you've been keeping up in your reading. Right? Well, I hope you've been keeping up in your reading. Otherwise, you got a hell of an all-nighter ahead of you. Okay? Well, so the only answer there that makes sense is this. You just prepared an aqueous solution. That's simple. You've just prepared an aqueous solution. This is incorrect, right? I mean, the very nature of sugar is that it has those nice electropositive and electronegative regions. Lots of OH groups, which are polar, which interact with the water. Okay, instant coffee does the same thing. Okay? So the answer is D. All right, number 10. So we're looking for an analogy for the way electrons move around the nucleus of an atom. This is actually like one of those dumb SAT questions that everybody hates. where, you know, A is to B as C is to D. Didn't you hate those? I did. I hated those. I did good on them, but I hated them. In fact, most of those answers imply linear movement, right, rather than rotational movement. So they must be incorrect. If you had linear movement, you'd have this electron that sort of moves, interacts with the nucleus, and then goes on its merry way. Doesn't work. They must be staying around the nucleus. Answer C implies no motion, and we know that's not the case. So it must be this one. This is the only one that implies rotational motion. And that's actually a good analogy because, as I said, electrons don't have any angular momentum. It's because they don't follow in these nice established paths. They sort of whiz around within an orbital, and they're sort of in many different places within that orbital. Okay? So it really is like a moth flying around a bright lamp. It's sort of meandering around rather than following a set path. Okay, this next one, number 11, should be a gimme as well, I hope. Should be easy. Well, let's name these just for yucks. We've got a methyl here, right? We've got a hydroxyl here. We've got a carbonyl here, and we've got a carboxyl here. And yeah, this is an amino. All right. B is the correct answer. The NH2 is the amino group. Okay. If you don't remember that, just try and remember amino acid. Right? Amino acids have an amino group on one side and carboxyl on the other. That's what makes them acids. This AMI, that name, that's in organic chemistry, it always refers to nitrogen. All right? So we always have nitrogen there.
So what's a good way to recognize that that's an acid?
The third way to recognize that what's an acid?
No, what's the best way to recognize that that's an acid?
Oh, that this carboxyl is an acid? Because the amino is not an acid, it's actually basic. Here's the way it works. We have an NH2, okay? We have a carboxyl. This hydrogen is being pulled away both by this oxygen and by this oxygen. And so what happens is this is easy to pop off. And so it does. It pops off into solution. So you get a C double bond O minus, and you get an H plus in solution. You've added a hydrogen ion, pH went down. That's why that's an acid. Okay. It turns out that the NH2 is a base, however, because hydrogen ions can come in and interact with the nitrogen, and that gives you an NH3 plus. Gives you a plus charged side group. Okay? So amino acids in solution are actually COO minus on one side and NH3 plus on the other. Right? So they have charges. Now that's why this is an acid. These acids are tending to drop off hydrogen ions into solution, and so that's why this is an acid. Why doesn't the nitrogen lose these hydrogens? Because it's not as it doesn't pull electrons as well as oxygen does. Oxygen is a little bit bigger, and therefore it's able to pull away electrons more effectively than nitrogen is. That's why it doesn't lose the hydrogens. It's a good question. Okay, number 12. Protein containing more than one polypeptide chain exhibits the blank level of protein structure. right, so to answer this question, you have to go back to chapter 3 and remember the different levels of protein structure. Primary level is just the linear sequence of amino acids in the polypeptide. Secondary structure is the formation of hydrogen bonding in the backbone of that molecule that produces the alpha helix or the beta-pleated sheet. Those are secondary structures. Tertiary is what happens when you get side chain interactions so that the protein folds up on itself and forms this nice three-dimensional structure. Okay? So that's tertiary. Quaternary is when you take two or more polypeptides and you stick them together by covalent interactions or by ionic bonds or by hydrogen bonds, hydrophobic interactions. All of those things are possible, but the idea is you're taking a couple of, two or more polypeptides and you stick them together. That's quaternary structure. So the answer here must be quaternary, a protein containing more than one polypeptide chain. Okay. In fact, you can have huge aggregates of proteins that form or polypeptides that form a single protein. Right. There are some enzymes that are like that. All right, number 13. How do you convert an oil into a substance that's solid at room temperature? So here you have to know how you do that, and you have to be able to justify your answer by providing the logic behind it. Okay? The answer is actually A. You add hydrogens and decrease the number of double bonds. Look on the back of an Almond Joy bar or any other kind of snack food that has oils in it. And it'll say partially hydrogenated whatever. Even potato chips have this. Partially hydrogenated soybean oil. What they've done is they've added hydrogens to reduce the number of double bonds so the stuff isn't so oily. You have more straight chain phospholipid molecules, and they pack better. So they're solid at room temperature, and they're really bad for you. Right? And I love potato chips. God, I could eat those things. I buy a thing of Pringles, and they're gone in 15 minutes. It's terrible. All right, but let's look at these other answers. Why aren't these correct? Removing water and causing a dehydration synthesis reaction to occur. Well, the logic is incorrect. You're not removing water from these things. You're not making a chemical change, really. You're just, well, okay, in a sense you are, because what you're doing is adding hydrogens and breaking double bonds. But on this one, you're not bonding those phospholipids to each other. Right? You're not sticking them together by covalent bonds. They're associating because they're able to pack in better. So this one, removing hydrogens and increasing the number of double bonds, that's going to have the exact opposite effect. That's going to make it more fluid instead of less fluid, because those fat molecules are going to be able to pack in even less. Yes, if you put it in the refrigerator, I would not be surprised if you could get it to become solid. But the logic is incorrect. It's not because double bonds form, it's just because the molecules are moving less, and so they pack in a little bit better. Okay, so I wouldn't be surprised if you could do that. All right, so that's why those other answers are incorrect. Now, before you go on to the last page, here's what I want you to do. Take out a blank sheet of paper. All right, what I want you to do, now this is going to be an honest-to-God quiz, but it's a bonus quiz, all right? Please do not discuss the answers with each other. This is to be your own work. What will happen here is that I'm going to have you answer the first 5 questions on that last page. Give your answers for them. I say this is a bonus quiz because this will be worth up to 10 bonus points towards the first exam. All right? It will not hurt you. It will only help you. All right, but please do not discuss the answers with your neighbor because this is to be your own work. All right? Discussing with your neighbors is academic dishonesty. So go ahead and make sure you write your name and your full student ID number across the top of this blank page, and then give me the answers for these 5 questions. There shouldn't be any talking. Please. Thank you. Hate to be anal.
So you might look at our...
Yeah, you can look at your sheet, of course. Just don't talk with your neighbor about the answers. All right, so go ahead and write down those answers.
Did you say last 5 questions?
The first 5 questions on that last page. Yes.
Am I done?
Pretty much. Pretty much, yes.
So I back up.
Yeah, you can.